Great. All right. Well, as Sarah said, welcoming you all to the Community and Citizen Science Collabinar. I know we probably still have a few folks joining bit by bit. This is only 12:03, but we'll get going. So for those who are new to the Center for Community and Citizen Science, we're a research center based in the School of Education at UC Davis, and we conduct research on various forms of community and citizen science in lots of different contexts and relating to lots of different environmental issues. And then we also try to help folks do that stuff and build toward different goals and work on different kinds of collaborations. So really excited about this topic today because it's getting into some issues that we're banging our heads against in some projects and that we know a lot of other folks are too. So that's going to be a fun conversation. Before we dive into the plan for today, I want to take a moment to acknowledge the land on which we're gathered here. And if folks would like to, in the chat, also acknowledge the land where they are, that they're more than welcome to. But for thousands of years, this land has been the home of the Patwin people. Today, there are 3 federally recognized Patwin tribes: Kachildehi Band of Wintun Indians, the Calusa Indian Community, Quetzaldehi Wintun Nation, and Yochadehi Wintun Nation. The Patwin people have remained committed to the stewardship of this land over many centuries. It has been cherished and protected as elders have instructed the young through generations. And we're honored and grateful to be here today on their traditional lands. So with that, I will pass it over to Sarah, and she'll get us going.
Hello, everyone. I'm Sarah, and I'm the Community Education and Outreach Specialist here at the center. I'm just going to let you know a little bit about how this Collabinar event is going to run. But before I do that, just acknowledge, letting you know about our Collabinar events in general. These invite colleagues from across the globe to present their most exciting developments and challenges in the field of community and citizen science. Attending the Collabinar are people who engage with community and citizen science at different levels, as we're seeing now with all the introductions hopefully coming through the chat momentarily. So this 1-hour session aims to foster an environment of mutual learning, and it's really less of a formal structured webinar as it is a discussion on the topic or project brought to us by the presenters. So it's really back and forth, a lot of engagement. So throughout our time together, I'll monitor the chat where you should add your thoughts and questions, and we'll answer anything as we go along that seems pressing. But we'll have plenty of time at the end for discussion. So with that, I want to pass it over to MB, who's going to introduce our presenters today.
Let's see if we can hear.
Hello, you can hear me, right?
Yes.
Yes.
Okay, good. Okay, yes. So I'm MV or Melissa, if you prefer. And so I'm a postdoc here at I'm currently physically in Lafayette, but I also work up at Davis at the center. And I have a combined background in doing community-based research and also data science and statistical analysis. And so the fun thing about this is that Ben and Sarah and I all have a PhD advisor or co-advisor in common. And we all want to work on different sort of large community or citizen science generated data sets with sort of like trying to figure out what you can do with statistics to manage and use these data in good ways. And so Ben has been helping us with our marine protected area MPA watch data analysis. He's been advising us, and that's been really great. And Sarah and I have connected via a group called Data Science by Design, and we sort of kept discovering that we were all studying kind of the same things. And I was like, well, maybe we should all just like come talk about it together because that'd be cool. And so that is why we're here today. So I get the fun of getting to read the bios. So I'm reading your bios now. Ben Goldstein is a PhD candidate in ecology at UC Berkeley. His dissertation focuses on quantitative methods for species distribution modeling. He's especially interested in large-scale biodiversity data, including participatory science, and in understanding what these data reveal about both wildlife and human observers. Sarah Stout is an applied statistician with research interests in ecology and the communication of statistics. Stout received her doctorate in statistics from the University of California, Berkeley, and she is an assistant professor in the Department of Mathematics at Bucknell University. Should I do the Twitter handle? Do you want the Twitter handle up there? Follow Sarah on Twitter because she's awesome. And check out her recent book with Deborah Nolan, Communicating with Data, the Art of Writing for Data Science. So I think it'll be fun to chat about a variety of different questions about how you work with these kinds of data. On to you folks.
Awesome. Thanks so much. I think I'm going to kick it off and then I'll pass it to Ben in a few minutes. Thanks for having us. I'm going to try to do a screen share situation. All right. And I think that you could see most of this, if not the beautiful version, but this is just how Adobe is going to be. So I'm going to go full screen. Thanks again for having us. I'm going to kick things off. It says my screen sharing is paused, which I don't want. Hold on. Should have just made a beautiful PowerPoint like Ben. Okay, let's try again. We're gonna stick with this.
Yeah.
All right, see enough of this, right? I'm gonna stay right here. Okay, amazing. So I'm gonna start us off by thinking about, okay, we want to flip the script on community science, right? We want to think about what are we learning about the data collectors on our way to learning about the ecology. So this is, I'm going to be talking about joint work with, of course, Ben and our co-advisor, Perry DeValpine, and sort of how the evolution took us to work on this project. So as a statistician, I'm sort of broadly interested in thinking about what kinds of data we need to collect so that a wrong model will still be useful. So we know nature is really complicated. No matter how good a job we do about the modeling, it's going to be wrong in some way. And I want to make sure that we can still do good things with that model. And we know that we can collect data in a lot of different ways in ecology. There's presence-only data where I know where people saw things, but not necessarily where they were going about their day. We have presence-absence data where I at least know where people did and did not see things. And then when we're thinking about abundance, there's different ways of capturing sort of the animal. Do we mark the individuals? How are we going to do that kind of thing? So we're trying to understand. We know that it's expensive, it's hard to do. And so like, what can we get away with and what can't we? So some terminology just at a big picture. I'm not going to get into the weeds today, but like, first of all, what do I mean by useful, right? I want the data to contain some information about something we care about, even when the model is wrong, because statisticians are not a trusting bunch in general, right? I don't buy anyone's model, even my own. And so I want things to still sort of work when that happens. What we have to watch out for are observationally equivalent scenarios, which just means that two data generating processes within some model I care about look the same from the observable distribution of data, what the data looks like, even if unobserved parts of the data differ. So what do I mean by this? Think about two different scenarios. One where I go to a bunch of sites and report a polar bear on half of the sites. That could happen in two ways, right? I go to a bunch of sites, there truly is a polar bear on half the sites and I'm good at seeing them and I see them all, great. But I could also go to all the sites and there could be a polar bear on every site and I only see half of them because I'm inexperienced, right? Those look the same. I see a polar bear on half the sites, but they imply different things about the ecology. And that's where this sort of troubling stuff happens. In that situation, we have a lack of identifiability. I can't identify the average occurrence of that situation. And so we're worried about when two data generating processes within some model look the same, but imply something different about the ecology. Importantly, this is a function of the model. And so this is where things get gnarly, right? Like if things look okay in my model, but not in somebody else's model, what are we worried about? And so this is when I'm usually a downer in talks because I'm basically saying we can't say certain things with this kind of data, right? So with opportunistic data or data that's collected only once at a site when we think there might be imperfect detection, this is vulnerable to model misspecification. Again, I'm going to like wave my hands, but the idea is that things can look identifiable in a small model. And if that model is wrong, that identifiability doesn't outlast that model. So everything would be fine if we truly believed that model we're interested in, but that identifiability isn't guaranteed in a bigger model. And so I'm usually telling a story of like, we should be careful, maybe we shouldn't use this data for this type of purpose. And that's not always that's a hard sell. And so that sort of motivated this idea of, okay, we have a lot of this data. I can't just say we can't use it at all, right? Maybe it can inform things we're used to thinking about, but maybe we could think about a creative way to use this type of data and do something else with it. And that's sort of how this motivated the project with Ben. So what we did is we were thinking about different data sets. So Ben will talk a lot about eBird. I was thinking about pitfalls of iNaturalist data for certain things. And so we were thinking about, okay, what's the difference, right? Opportunistic data and iNaturalist, you say, oh, I see a squirrel here, right? I don't know my whole hiking path where I did or did not see the squirrel. But in eBird, we have a very systematic, rigorous checklist, right? Everything I see, I report, which means I know what I didn't see. And there's this joke, which maybe you all are familiar with, that like if you study birds, you're in luck because birders are very like type A people. They'll follow directions, like that data is good. And that also sort of motivated this project. Like, why is eBird so much better than iNaturalist? And wait, there are birds in iNaturalist, so can we do something with that? And so we decided, okay, what if we actually quantify that difference between the systematic data collection and the opportunistic data by comparing the birds in the two data sets? So we wanted to quantify this opportunism. This is sort of the motivation. And we tried to break it down in terms of how does a data point end up in one of these data sets? So first, the bird has to be there, right? That presence layer is what we actually care about, the ecology, right? We want to learn about birds, but we're distracted by the fact that like the bird has to be there and somebody has to detect it. So disentangling presence and detectability is a lot of the stuff that I'm a downer about usually, right? Like that part is hard. We have to revisit the sites perhaps. But if the bird is there, you detect it, it's definitely ending up in the eBird data set because you have to report what you see and you have that rigorous checklist. So we're done at the eBird level. But to end up in an iNaturalist data set, you have to go a step further. We're calling this the engagement layer. And it has to do with a couple different things like logistics, right? iNaturalist, you need a photo so that it can tell what it is. You don't need photos for eBird. And also you have to decide, yes, I'm going to take a picture of this thing, and yes, I'm going to upload it to iNaturalist. There's no protocol. You don't have to report everything you see. In fact, you won't because iNaturalist covers everything and you're not going to take a picture of every little blade of grass. And so that engagement filter is what we're trying to quantify. And it tells us the difference between how you end up in one data set versus how you end up in another. So we're going to try to measure that and put a number on. And so what does this data look like? So I have an example from Ben, of course. On the left here, we have eBird. It covers most of the US. There's lots of bird data. Again, birders are great. We can't hate on iNaturalist because you have to remember that iNaturalist has just much more going on, right? Not everybody's interested in birds on the app. So that sparsity sort of reflects a little bit about that. But we have the number of observations of, in this case, a bald eagle in each little region, out of all of the birds in that little region. And we're calling this a raw reporting rate. And we have this for many, many different birds, almost 500, basically the birds that we have enough data we feel like we can do something with. And so remember, we're trying to quantify the difference between these two things. And so the first thing we might want to do is like smooth this out and predict this reporting rate. Like, can we model these reporting rates that we're seeing raw here? Again, I'm going to do a little hand wave here and try to give you the gist, and we can talk about details if you want to later. But the idea is we smooth this out. We're modeling that reporting rate, which is just the idea of what's the probability I'm going to report this type of species as the next thing in this hex or this region, right? So like, I know how many birds were reported there. I want to know what's the probability that this one bird is the thing reported. And so the idea is we can do this for every bird, we can do this for both data sets, and we smooth across space so we get sort of a nicer picture of what's going on. We're going to do this in a statistically rigorous way, uncertainty quantification, all that good stuff. But for now, in pictures, what this looks like is just a smooth version of this, right? We have a predicted surface of that reporting rate. And now we can start to think about, okay, what we really care about is the difference between these two. We're trying to measure that filter of engagement or sort of logistics or charisma, whatever you want to call it. And so we do something pretty simple. We just take the iNat surface, we lay it on top of the eBird surface, we take the difference across each little region, and then take the median of those differences. We just want a typical difference between the two data sets. So we're smoothing over space, and we've already collapsed across time. This is just like a raw metric over the whole region. And for the bald eagle, we're not surprised that it's positive because if you see a bald eagle, you're going to be excited and you're definitely want to take a picture and brag that you saw it on iNaturalist. It's also big, so you can actually take a picture from far away. So there's all that sort of helping us definitely spot that bald eagle in the data set. Now, this over-reporting index also comes with uncertainty, all that good stuff, but we're going to have one of these now for every single bird in the data set. And now when we look at this together, we have a lot of information about what that bird data actually looks like. Because remember, eBird goes into statistical models, so does iNaturalist. And so what we know is that about 150 birds are underreported in iNaturalist compared to some gold standard eBird. About 50 are overreported, and then the rest are sort of not significantly different. But when we zoom in and look at these, it sort of confirms what we expect. We see the Indian peafowl, that's a peacock, turkey, owls, herons, of course, the bald eagle, all overreported. That checks with our understanding of like, if you saw that bird, you'd be excited to snap a picture of it and report it.
Sarah,
And then underreported birds are maybe birds you see all the time. Once you've seen one crow, you've seen them all. You're not going to snap a picture every time you see one. And so that also checks out with our sort of like gut feeling about what's happening. And what's important now is like, we all think about this when we look at the data, but now we can put a number on it. And if we're using these things in models, this helps us quantify those things we know intuitively based on our experience with the apps ourselves.
we have a question.
Oh, sorry, yes.
How'd you do that? What'd you do? Thank you. Hi, I just really wanted to quickly ask, so that I can track what you're saying on this slide is reporting which app compared to which. Is this INAT only with respect to eBird or with respect to what we know about bird data in general from other sources?
This is just INAT versus eBird.
Ooh,
So eBird as like a standard, maybe it's not
Just think that
the complete picture of what birds look like on the ground, but we're saying like,
interesting.
So there's the assumption about eBird is that it is a representation of bird abundance, bird reality that is much closer than iNaturalist. So the difference between those things is meaningful when you, in terms of understanding what people do on iNaturalist. Is that
right.
we're not learning about what eBird observers do here, really. What we're learning about is INAT
In
with
relationship.
with reference to eBird.
Yeah.
Exactly. Yes. We're mostly focused on those opportunistic birders. We're thinking that the typical eBirder is doing something more rigorous. And so we want to understand the more loosey-goosey data collection going on.
And can I add something there? Oh, sorry, go ahead.
Later, oh, Ben, go ahead.
No, please.
I was just gonna say later at the end, I just want to ask about how you determined that. So I wasn't sure, there's a little bit of that I'm still struggling with about that. So I'd love to know how we know that for sure that eBird data is closer to reality. I mean, I know that birders are better than iNaturalisters by definition, but just wondering, so I see like if you say like
Is this being recorded?
that's the bar. I know, right? That was probably broader than I meant, but you know what I'm saying? Like, I'm just, because I know I submit stuff to eBird all the time, opportunistically, because I saw something charismatic and moving, and that was what I submitted. Like, it was not systematic or rigorous in any way. So and Ryan says he does that too, but we could be weird. And I just wonder if there's data about that, like, because there's the iNaturalist bar, there's the eBird bar, and then there's the actual reality observed by professionals in a rigorous transect or whatever kind of way, right?
Right. I should note that in eBird, you can tell it whether you did a complete checklist versus if you did something. And we are using the complete checklist.
Yeah.
So that's the
Gotcha.
comparison. You're eliminating all the opportunistic blah blah blah.
Yes.
Because that's important to say. That's great to know. I'm so glad that, because I was like, I know that there's a lot of not effort reporting that happens on eBird. So got it. Thank you so much.
Yes, no, glad to clarify. Yes. So like most of the data on eBird is actually in the complete checklist bin. And so we're focusing on that as sort of the closest we can get.
The bar, yeah.
Yeah.
Can I ask
Ben, were you gonna say something?
a clarifying question
That's
about
good.
this slide? When the word overreported is overreported with respect to eBird,
Yeah,
right?
both of those. That's
Okay.
what I was.
Gotcha. Okay, so but it's the relative amount of, say, bald eagles in an INAT data set is is much higher than you would expect, given all the other birds in INAT. Okay.
Mhm, mhm.
That's exactly right. Sarah, would you put the like conceptual diagram back up real quick?
Yes.
I wanted to make a comment about this. So like one of the sort of starting point from this was the realization, so, so I do a lot of species distribution modeling with all sorts of data types. And what we're interested in there is differentiating between true presence and the detection process, right? So we kind of started from this place with the iNaturalist data of saying, we can't do that. We don't have absence data.
Right.
It's not possible to differentiate between presence and detectability. But what we realized was the eBird data, we think, represent the combination of true presence and detectability. So if we're willing to make the assumption that the interesting parts of detectability and presence are the same in the two data sets, right? Like no matter which data set you're working with, if you're an expert and if you don't have opinions about which birds you're interested in, you're going to see the same birds at the same place, such that any difference we claim is attributable to this engagement process, which we think is something that we can separate out from presence and detectability.
Got it.
That's fascinating.
All right.
Thank you. Sorry for the interruption, but that
No, that's good.
I was stewing in my juices
Yeah,
here.
no, that's good. When you're in it, you forget all these like important details.
Yeah, I
Um,
really did that yesterday with the COB meeting.
okay, so we learned about the bird data, and now we're going to take it a step further and think about what does this tell us about the birders? So we have all these over-reporting indices, and we want to connect those to traits about the birds. Can we predict your over-reporting index based on what the bird looks like or how common it is? And so now we can flip it so that the over-reporting index is the response, and we're trying to understand that. So we're going to now do a meta-analysis of that with things like the mass of the bird, the color, the range size, the rate at which it appears in these checklists. And so like these are sort of just an exploration of that data, but you can picture that a model is going to synthesize all the things you're seeing on the screen. And now like, can we boil it down to like, is one of these a significant predictor of your over-reporting index? And so the takeaway is yes, right? So we already sort of quantified our intuition that some things are more over-reported and they're going to be big, exciting birds. And some things are going to be underreported that are annoying or common birds. But when we do the meta-analysis, there is a positive significant effect of mass and color. So bigger, more colorful birds are more likely to be over-reported. There's a negative significant effect of the number of hexes where the bird is reported. So think about that as a proxy for range size. If it's everywhere, like you've maybe seen one, you've seen them all. And then in the presence of all that stuff, the proportion of checklists where it's reported, maybe a proxy for abundance, is not significant, but we're sort of not surprised by that because it's probably highly correlated with range size. And so again, like maybe we're not surprised by any of these findings, but being able to sort of quantify that this is really happening is going to be helpful when people are thinking about how we use this data. And so now we have this more complete picture. We can use this for conservation, right? We know about the data collectors themselves. We're also thinking about what else does that data tell us about the people behind the data, not necessarily the animals they're focusing on. But you can imagine tailoring conservation and outreach if there's some unsung bird in your area, right? Why is nobody paying attention to this bird? Like now we know that information. Of course, as a statistician, I care about the modeling implications of this. So we're thinking about, okay, can we use this index to calibrate between the two data sets? There's all this interest in data fusion techniques, bringing all the data together. This metric now informs the difference between the two, and that might help bring them together in a smarter way. We're also thinking about using this approach in a more targeted region or time period to tailor the indices. So we've done a lot of smoothing over space and time here, but you can imagine redoing the whole sort of scheme on a targeted region or maybe making it temporally explicit. We're doing that right now to sort of think about how there's a one number summary, but there's also a lot of variability we haven't explored yet. And then of course we can do this for other animals because there's other systematic data collection out there, not just for birds, and iNaturalist has everything. So we should be able to do this similar comparison and try to tease out other things about other species. And with that, I want to pass it over to Ben. I just want to flash some other things that I care about on the screen before I do that. Other things you might want to talk to me about later, a lot about communicating, writing about data, how do we teach that kind of thing. But for that now, I will pass it off.
Sarah, can I actually suggest, so my plan was to present, and still is to present, a sort of quick overview of a different project where we're applying, sort of using eBird to do species distribution modeling. But maybe it's a better idea to do Q&A if people have thoughts on this project before I sort of take it in a totally different direction.
Sure, and I can stop sharing so we can both sort of.
We like that idea.
Yeah.
Cool.
Yeah, what questions do you have?
How about folks from Zoom? There's a lot of you, more of you on screen than in the room. So folks from Zoom, questions for Sarah? I know, me too. This one, holding back.
I think one of my questions was going to, I think sort of pinging on what Heidi had said before about the assumptions of behavior of eBird versus iNaturalist. Is there, or has there been any conversations also about the age of the various participants? I'm just thinking, you know, a lot of eBirders started doing eBird when it was still paper pencil. And does that make a difference in how strict to the protocols they stay?
That's interesting. And I'm like, immediately like, how would we know how old they are? But we do know,
We know a lot.
in principle, like by user, you could see how long, like when's their first submission, when's their last submission.
Could it be experience on the site? It might not be age, like the actual biological age of the human, but it could be time on the site, right?
Right.
Well, I think that it would imply a little bit of age, only because like initially, like when the site first started too, you were still doing everything paper and pencil, and then we still had to physically go over to a computer. You know, the app wasn't a thing. So if you had somebody who had an account that was pre-eBird app, not just experience wise, but also like when they learned those protocols.
Right.
Yeah, and that's definitely the type of thing, like the metadata behind the actual observations could be mined to try to like, how do we quantify that? How would could we split the groups up? What would be the metric we'd compare them on? Like who becomes the gold standard? Like all of this, we have to pick something to be relative to something else. But that's a great idea of like, and exactly to the spirit of like what we're trying to do is like, maybe we're focusing so much on learning about the birds that we forget that there's like people behind all of that. And like, what does the data tell us about them in a non-creepy way, I guess.
Right. Yeah, that's a fair point to not be creepy. Oh, I'm sorry, Gary had his hand up. I was going to ask about CBC and BBS data, but Gary, you go first.
Okay, thanks. So Sarah, thanks for a lovely presentation, some really awesome data and ways of leveraging these community-driven data collection efforts. I'm interested in knowing, like specifically for California, your last slide prior to your other interests talked about calibrating for specific regions. And I'm really interested in that. We're currently working on, through the Natural Reserve System and with CDFW, California Department of Fish and Wildlife, a program to develop what we're terming a biodiversity sentinel site network. If you haven't heard about it, I'm happy to fill you in more, but the gist of it is we want to start with bird data. And I had seen you and Ben's paper, and I'm really interested in what you think, just off the top of your head, or if you've done any preliminary analyses of how the patterns you shared in this presentation might differ at a regional scale with, say, just California.
Yeah, and actually Ben's probably better to talk about this because he's more in the eBird at the California scale. So I'm gonna punt on it. And Ben, what do you think?
That's a great question. I have not done this analysis just for California. I've worked with the eBird data in California. So the project that I'll share in a minute is in California with actually some folks at CDFW and also Danny Carpet Davis is on that project as well. But, you know, I don't know. So, okay, so I can share that we've done, Sarah and I are in the middle of a collaboration with some butterfly researchers where we're looking at, there's a program called eButterfly. We're comparing that to iNaturalist. And in that project, we're comparing sort of East Coast, West Coast. And at the species level, very preliminarily, the patterns seem kind of quite similar in the big picture, as in monarchs are popular on both coasts, cabbage whites are unpopular on both coasts. So my guess is that more often than not, it's probably region generic, but that's totally speculative for California.
Thanks.
Yeah, Gary, if you have a follow-up, but I have a question. I mean, I think this work is totally fascinating. It's a completely unexpected angle into a question that I've long had about iNaturalist, which is a sort of anthropological interest in like what motivates observations at various points. And I don't know if it's possible using your approach to get into some of these other things, but it occurs to me that as an iNaturalist user, I am motivated differently at different times when I make observations. In my garden, I'm like, is this a pest? And, you know, out on my walk with my dogs, I'm like, oh my God, it's a bald eagle. I can't wait to tell everyone about this. So is there, and I always imagined you'd have to just do some sort of super elaborate survey or interview project to really sort of get at that. But it just occurs to me that you've gotten it at something that in some ways it's like, yes, the sky is blue. Yes, charismatic megafauna are overrepresented on iNaturalist, but are there ways to get at some of the other dynamics that structure iNat data outside of just that?
Yeah, I think definitely, like we've explored, so, okay, backing up, like it's hard because on iNaturalist, there's such a dramatic, probably on eBird too, but there's a dramatic difference in like people who use it very infrequently and then people who use it all the time. And so like the power to detect any like real effect, we can only really do for the most popular users, but then they aren't representative of this like thing because they're probably scientists or they're probably like really engaged and they're thinking about these things more. So it's like a little bit tricky there. But I know that there are some papers who do these surveys of users that try to like get in their head of like what causes you to open the app, like, or to start a checklist, even in eBird. And I think that one thing I'm interested in is like, okay, so if we have all these surveys, that tells us what to go looking for, and can we convince ourselves that we find it in the data? Like what's the relevant matchup? Because I think you could go searching for anything, but the survey gives you like something to search for and then try to like quantify that and then go from there. But I think there's a lot of interesting like slices of the data. It's just sort of like getting crafty about like, what would this look like? How do I look for it? And like subsetting the right groups of people where you could even have a hope of detecting it.
Can I'm conscious of the time. I realize that we want to give Ben his time.
Oh yeah, Ben.
I just wanted to really quick flag, like there are at least one, if not more, social science researchers at the Cornell Lab of Ornithology that entirely study the behaviors and the motivations and the learning that are happening, right? From eBird and other bird projects at Cornell, but then also, I know that there's a lot of learning research, including mine, on like motivations of iNaturalist users, right? And like what they're getting out of it or what they're doing it for in the first place. In my case, it's kids, which is not the kind of subtleties I think that Ryan's talking about, but there's a lot out there in the I would just suggest looking at the learning and education world for that, because there's tons of people that are doing interviews and surveys with iNaturalist users. But the key I was going to flag is, about, and I could find the article, but I know Julia Parrish and her team at Coast has done a lot of bridging that you're talking about, basically linking the surveys and the interviews and the focus group data with users, with their data collector humans, right? And the accuracy and the frequency and the duration of their data submissions, right? Especially accuracy. So that's something you could look at. Like they did all kind of fancy. She called them the Wunderkind. She had a postdoc just like doing that analysis to link those two things in a really fancy modeling way. And so, and I can't remember his last name. His first name's Tim. So Parrish, have you seen some of those? Have you seen the articles I'm talking about that link the data about accuracy and duration and like when they joined and how long they've been retained in the project and stuff like that?
No, but I'm gonna immediately Google Scholar that.
Well, I can
before
find it
forget.
for you. I just wanted to make sure I wasn't telling you something you already knew. So
Yeah, no, that sounds great.
I will find it for you.
Awesome, thank you.
But before we completely shift gears, I had just wanted to throw in there the idea that we're talking about like what could you sort of extrapolate from just the data of that person. So like, for example, how long and they've been in that database tells you something about how long they've been doing the sampling. And I mean, like one of the kind of canonical like examples of working with citizen science data on birds is the breeding bird survey and Christmas bird count stuff that Lincoln Sauer did. And I think they were looking at that as they were modeling like observer effects where they thought, my understanding, I have to look at the paper again, was that like the idea is that your individual count would go up over time just because you like got better at counting things. And so there was a way to kind of like assume a functional form for that and put it into the model. And so that kind of made me think maybe there would be a way to kind of triangulate between or synthesize between something like that kind of data where there's overlap between that and eBird and iNaturalist. And I don't know, it becomes kind of a monster after a while of like, there's 5 data sets here, but you know, hierarchical models are, you know, always a fun time, as we know. Anyway, carry on.
That's a really good point. And I know they've done that with eBird. I know the eBird folks have shown, you know, if you include sort of how many checklists have you already submitted, it, you know, improves the model in a vague sense. Yeah, I will say I really appreciate the chance to talk about this project with this group of people. I mean, you guys seem really, you know, Sarah and I, well, I won't speak for Sarah, but I spend most of my time around, you know, ecologists who aren't thinking about the data and don't tend to get as excited about sort of the observer process as Sarah and I have gotten through this project. So yeah, thanks for plugging us in, MV, to this community. I'm gonna do a really quick overview of an applied project that is ongoing, but hopefully mostly wrapped up as part of my dissertation. I'm going to try to be really brief so that we can keep the conversation going. But broadly what this project looked at was sort of what does eBird tell us about drought impacts in California's Central Valley, a region that you all know more about than I do because you all live there. So let's see. Oh yeah, so these slides come from a talk that was more about the ecology side of things than the data. So I've gone through and I've put these gold stars in places to remind myself to talk about weird eBird data things that we had to address with modeling. So for, you know, conservation background, I don't think I need to spend much time convincing you all that drought is an important issue for conservation in California. And it's a complicated ecological process to understand and predict from. We know broadly that birds are sensitive to drought all over the world, but we don't really, we have a wealth of hypotheses about how drought impacts species, whether it's directly through sort of extreme heat, lack of water, or more indirectly through changing vegetative productivity or sort of trophic impacts. And we also are starting to suspect that drought impacts might be really variable in space and time. For example, individuals of a certain species that may have an overall sensitivity to drought might do better in certain habitats. So really characterizing all these things, we thought at the beginning of this project, you know, it's going to take a data set where we have a lot of data at high spatiotemporal resolution over a big region with a lot of habitat variability. So we became very interested in eBird for addressing this. Looking at the California Central Valley, super interesting region for birds, you know, important migratory corridor, also important breeding ground. And as we'll see, it turns out that human modification of the landscape is a really important ecological process in this region. And luckily, where there is extensive human modification, we also see extensive participatory science data. So there's so much eBird data in California, which is kind of an amazing resource if we can figure out how to leverage it. So we were looking specifically at the Central Valley. The first thing we did was we followed the guidelines for basically how can we take this enormous data set. I think after restricting to our spatial and temporal extent of interest, which was the breeding season of 2010 to 2019, we had something like 55,000 complete checklists that we could be selected with. So we were able to filter extensively, for example, only looking at stationary point count surveys as opposed to walking transects, filtering for sort of maximum duration, maximum observers in the group, thanks to the sort of availability of all this sampling metadata that you get from eBird. So we ended up with 15.5 thousand observations that after we'd filtered for quality. So we were able to be really aggressive and still retain a decent amount of data. And you see, the number of checklists increasing over time, but even back in 2010, we had a decent amount of coverage across the region. And we identified 66 species that we were interested in, basically the 66 species that were the most common and appeared on at least 500 checklists throughout this period. So to get at how a bunch of landscapes, so habitat type and climate processes influenced bird abundance or reported counts, we used single species and mixture models. So and mixture models are basically a hierarchical extension of a count model that's attempting to differentiate between a detection process and a sort of true abundance process by making some assumptions about sharing information across replicate observations within like a unit, an ecological unit. We stuck a bunch of statistical structure on the sides of these things. So we allowed for 2 dimensions of overdispersion, and we put in site level and also observer level random effects on abundance and detection. And we included a bunch of sort of eBird eBird metadata in the detection process of the model. So the idea here was basically, we have such a huge amount of data. Let's be as defensive as possible in allowing heterogeneity in the data to come from anything other than the variables that we're interested in. So that we're not sort of making incorrect inference. Here's a sort of schematic flow chart of the mixture model. And we ran into this trade-off between sort of 2 paradigms of modeling drought. On the one hand, we're interested in breaking drought into these sort of different climate processes, wetness, vegetative productivity, temperature, precipitation. But on the other hand, we wanted a sort of a dial that we could turn on the entire model to look for an overall impact of drought. And especially in light of the fact that a lot of these climate variables are really correlated with each other. So what we ended up doing was this giant multi-model framework where we use linear mixed models to predict our climate variables as a function of drought and habitat type. And then we use observed climate variables and habitat type to predict abundance. We smash those things together and do this giant Bayesian posterior predictive workflow to basically say, if we set our dial to drought, what overall abundances do we predict for each species? If we set our dial to non-drought, what do we predict? And then how do those 2 compare? So what we ended up with was predictions of the change in overall abundance for each of the 66 species. We found moderate patterns that sort of agree with the literature. So 16 of our 66 species credibly decreased and 5 had positive associations with drought in the region. One thing I want to put a pin in this question of overall drought to discuss as a group is one of the major limitations of this type of study is that we didn't systematically select where eBird sampling happened. So our inference is really restricted to a subset of the study area that represents where eBird sampling actually took place. So, at the end of the day, we can really only do inference about where eBirders have chosen to go, which is, I think, an important consideration in light of especially claims we're going to make about habitat type. So we had the second question about what's the role of habitat during drought, and specifically we wanted to know is the impact of drought on abundance mediated by habitat type. I'll skip over this. Broadly, what we did was we derived an interaction term, sort of did some math to connect different components of the model and came up with a number for each species representing whether or not the impact of drought depended on habitat. And we found that actually far more of our species had habitat associations with drought than did change in their overall abundance. And we saw some pretty consistent patterns across species in terms of what the changing habitat associations looked like. So this ternary plot on the side, if you follow from the black dots, the non-drought condition, to the white dots, you'll see that most of our species move away from natural habitat during drought and have a greater rate of use of especially developed, so urban areas, and then also specifically perennial agriculture, so vineyards, orchards. And what we think is going on here is that during drought, species are moving from natural areas which are drying rapidly and into areas that have sort of human, artificially human-induced irrigation, and also have sort of microhabitat structure. So we think probably trees are really important, or canopy, for helping birds survive extreme heat. We also looked at how did impacts break down across our different variables, and we found that basically precipitation and temperature were telling the whole story. So our wetness and greenness indices turned out not to be important at all, according to our model. So specifically extreme temperature seems like the main thing that's driving bird abundance during drought. So to sort of synthesize at the high level, we think habitat associations are really important. We think temperature and water availability are really important. And we suspect that human modification of the landscape, and especially irrigation, are going to be, big pieces of how we see birds responding to drought in the future. And of course, a study at this scale was really only possible thanks to the availability of the eBird data. So I'll leave it at that for now. See if there are any questions. Or if folks want to move back to the other.
Chris says bees do the same thing. Bees,
oh, interesting.
Yeah, they urban areas can be like reservoirs for native bee populations during times of drought because there's so much more floral diversity in cities because people water their gardens essentially.
Yeah, that makes sense.
I just want to make a point to all of my colleagues at the Center for Community and Citizen Science that I have spared you slides that have distributions on them, but I've been doing exactly that same stuff. That was not a criticism of Ben. I loved that slide. I was like, oh, random effects. But that's that you guys haven't seen me, I think, get like fully gleefully statistical. So I have loved getting to see that. Thank you for sharing that.
Well, and I can say that I appreciated Ben's explanation of those slides. And so if and I feel like he spoke about them not in a gleefully statistical way, but more communication skills. So we can handle it, right? I think we can handle it.
All right. Okay. All right. I'll practice my Ben-like and less gleefully statistical ways of talking. Yeah, Ben, that was a great explanation. Thank you.
Well, it's good.
Thank you.
Definitely gleeful. I don't want to harsh your mellow on that.
Yeah. I think you were talking, and I may have just missed some of what you were saying about the distribution of eBird observations in this geographic region and what constraints that created for you. I'm curious about sort of the realities of density of eBird observations and what that meant for this analysis. And sorry if you already explained that.
No, that's a great question. Yeah, so first of all, it will not come as a surprise to anyone that the urban developed land cover category was very oversampled in the data set. So way more of our observations came from population centers than were sort of representative of the landscape, which is not in itself a problem in the sense that, that's something that we're sort of accounting for in the model. So in theory, if our model is right, we don't need to sample things evenly in a spatial sense. I think where I become more concerned with, making general inference from this model is what are the ways in which sampling is non-representative, but also not captured by the covariates that we're thinking about. So, for example, are eBird observers really sampling developed areas, or are they sampling parks in cities? That's something that we didn't put into the model. Are they really sampling, the middle of a bunch of, row crop fields, or are they, at trails or, along roads on the sides of those fields? And then the other issue is, what's going on in the holes in the data set? Is it fair to use the land cover types that we've identified to extrapolate across the study area? Or, for example, the northern half of the valley is sampled with a lot better coverage than the southern half, and those are two pretty different ecosystems. So that's not necessarily something that we can capture. So I think our high level patterns, I think it's, we've accurately sort of summarized the data that we have, but I would hesitate to, start implementing policy based on this study without sort of a follow-up to sort of check these hypotheses.
More research is needed.
More research is needed.
I don't, if there are other questions, I'm gonna.
Yeah. So, do you want to unshare your slides so that everybody can see, and then maybe if there's anyone on
It looks like Gary has a question.
the Zoom land. Oh, Gary, great.
Hey again, thanks for a nice presentation, Ben. And I'm curious, your covariates that you used, you showed on that slide that you had greenness, wetness, temp, and precipitation. And then did you have like a combined covariate of temperature and precipitation? Like that one slide showed both. So just curious if, what
That's
amount of variation did it explain actually? And do you think there's room for others?
Yeah, so what we did was, I didn't really talk about the details of this, but the principle behind the posterior prediction is, use covariates representing one condition from the linear models to represent drought, use covariates representing the non-drought condition. And then for those sort of one variable variation bar charts, basically what we're doing is we're saying what happens if we make predictions but change only precipitation to its drought condition. So the temperature and precipitation was change temperature and precipitation to a drought condition, hold the others fixed. And the predictions from that scenario were like identical down to, I think, maybe one or two species with the full scenario. I have some thoughts on what's going on there, but I don't really want to get into it. We can email about it if you want. Josie, did you have a question?
Yes, thank you. Both of your talks were fascinating to me because I'm relatively new to CCS and I'm trying to think about how I can use it for what I study. I'm really interested in urban animal behavior and how animals interact with people. And so as folks that have worked with both of these data sets, either eBird or iNaturalist or others, do you have any thoughts for metrics that we might be able to get out of this? This is kind of speaking to that metadata question on how humans are interacting with animals. That's a broad question, but
no, I
didn't want to make it more specific or narrow. I wanted to see what your thoughts were.
I think our sort of engagement index from Sarah's presentation could be considered a metric of human engagement with birds. So in the sense that like we basically can hand you a number with uncertainty that represents the extent to which we think people are more likely to interact with that species in such a way that, you know, it ends up on iNaturalist relative to how often they would be seeing it if they were species agnostic. Does that sound right, Sarah?
Yeah, and I think also like the more the behavior you care about is like weird, the better the data actually is. Like, so if you're like, I know that this is going to seem like a tangent, but I'll bring it back. So like there are these researchers at Penn State who I know who study like there's this contamination of the specific flower, and you can tell if it's contaminated, it's like some fungus or something. Yes, that's this very scientific explanation. But the important part is it makes the flower look more colorful. And so actually, like on iNaturalist, if you take a picture of it, you can see whether it has that fungus or not. And because it makes you like notice it, they actually have great data because it's like playing into the like engagement or opportunism or charisma of the thing. And so if you're thinking about like other animals in the urban environment, if they're doing something that would like be, oh, I have to document that, that's actually like, you can like lean on the bias to actually get more data for the thing you care about. And maybe I'm misinterpreting what you mean by like the interactions, but I'm just thinking of like, I don't know, this is stereotypical, but like your charismatic raccoon who like is always knocking over your garbage can or something like that. And like, are there more raccoons? And you have pictures of them and you can, are they distinctive by their, now I'm like straying into ecology territory and I'm not an ecologist. But anyway, does that ramble?
Yeah, no, it makes sense that the behavior that's more salient will be more likely to be recorded, or like is of more interest to participants to record. I was thinking also of like, maybe if there was metadata on like, I don't know, if there's photos associated with these observations, if there's information on like the camera and the zoom lens that could get at actual physical interaction. Like, are you 100 meters away, or are you with your iPhone kind of 5 meters away? But I don't know if that's
there's definitely
idea.
data on that.
Okay.
Yeah,
Sorry,
that's a free project idea is, can you use like the resolution of the iNaturalist image to differentiate between like wildlife photographer behavior and someone snapping a photo on their phone, you know, to be like, hey, what's this species?
I'm going to suggest, just because it's after the time and I just noticed someone dropped off, that there's so many juicy conversations that the people on this session might have. If you'd be interested in having more conversations, you could put your email in the chat, and that would let people like, I don't know, say Josie pick the brains of, I don't know, people like Sarah and Ben and Gary about this stuff. But you know, brain picking can happen across all the ways. So Ryan, do you want to wrap us up or anything like that?
I suppose we need to wrap up. I'm sorry. I have lots of questions too. This is really a fantastic session. Thank you both so much for spending the time and having the discussions with us and presenting the work.
And for new people or people we haven't seen in a while for joining. So it's a cool thing that it brought. We've gotten a really cool mix of people in this. It's really cool that Ben, to your point about cross-pollinating across just like really different disciplines, honestly, but we're all interested in this kind of same thing. So really cool. Thank you very much, everyone.
Thanks so much for having us.
Yeah, thanks. We'd love to hear from you. We're always noodling on this kind of stuff.